Okay, we're going to get started, and our first speaker today is Natasha Kaplan. She got her PhD from the University of London, King's College Hospital Medical School. Then she did postdoctoral training at St. Mary's Hospital Medical School, Imperial College. And then in 1996, she joined the National Human Genome Research Institute, and NCI was smart enough to hire her in 2004. So now she's in the genetics branch, and she's going to talk to us today about functional genomics. Natasha.
Thank you. Thank you very much, Terry. I feel like this should be a tutorial. We should have everyone a little closer, but maybe you all want to stay over there. All right, so what I'm going to do today, get started. Everyone can hear me okay? Okay. All right, so what I want to do is start off with a brief introduction. We're going to do some definitions. So some of these slides are going to feel a little wordy, but it's really just to make sure everybody's on the same page about what we're talking about. Then I'm going to move into technology. I'm going to be focusing. There. Oh. No. Okay, there we go. Sorry. All right, so my introduction, we're going to start with some definitions, and then we're going to go into technology, which I'm going to focus on loss of function analysis. And then for at least half, if not slightly more, I want to actually give you some real-world examples of how my lab and lab collaborations that we've conducted have applied some of the technologies that we've used, either for single gene analysis or for screening-based analysis. Okay, so some definitions. Why? Why do we need functional genomics? What is it that? Why do we need this? So I think most of you are aware that obviously being able to profile on a genome-wide basis, on a transcriptome basis, DNA copy number, variations in sequence, mutations in sequence, DNA and chromatin modifications, and then actually being able to profile gene expression data, that these very large-scale data sets have absolutely revolutionized how we understand the disequilibrium that's underlying cancer and other diseases. The problem's really been functionalizing that information, trying to make that link between what we're seeing as a disease change, whether it's a mutation, and how that's actually affecting the function within a cell and how that's leading to tumorigenesis in this case. So we need mechanisms that will allow us, or need technologies that will allow us to functionalize on a similar scale to what we can do for other methods. So the Oxford Dictionary of Biochemistry and Molecular Biology defines functional genomics as the systematic analysis of genes, their encoded proteins, and the relationships between that gene activity and cell function at the level of whole genomes. Now, that in some cases can actually mean that you're looking at just one gene and then looking at how that's affecting an entire genome. Or, as you'll see at the very end, it could mean that you're doing systematic analysis across the entire genome. So it's sort of both ends of that. Most of you probably are doing some element of functional genomics, if you like. It just, in this case, is some of it's a mixture of scale and a mixture of how you're just looking at the data. So I'm just going to have one slide on gain of function, but I did want to introduce that just in case some people are interested in this. This is used still widely. More widely is loss of function analysis, but it is still there. I think it's still something that people need to be aware of. And this is a review that sort of put together most of the features that we talk about, or how overexpression of a wild type protein is usually, or a mutant protein is usually used. So you can overexpress a wild type in a mutant background to do a rescue experiment to look at the function there. You can overexpress multiple different proteins and look at these their interactions in cases of epistasis. You can use it for screening for different drug targets. There are lots of different contexts where this is used. But this is usually less done systematically. There have, in some cases, been open reading frame-based screens done, but they're much rarer than the loss of function analysis. So it's really that I'm going to focus on, and I'm really going to focus on two flavors of loss of function. That's where you are disrupting normal gene expression, and that's really is still used as the gold standard for trying to assert what is the function of a particular protein, or more seeing increasingly the function of a particular non-coding RNA, maybe a long non-coding RNA. And that's where you're doing either a gene knockdown, which is actually a phrase I don't particularly care for, but we sort of is ended up in the literature, where you actually have a reduction in the level of the gene expression and therefore the amount of protein there, versus where you've got a complete gene knockout. There is now no protein being produced. And these can give us very useful phenotypic differences that allow us to really understand how a particular protein is working. So there are other two terms that I just want you to be aware of, that we've actually been doing these types of loss of function analysis really now for many decades. But in that case, it's what is usually referred to as forward genetics. And that's the mapping of a phenotype to a mutation where there's actually no prior genetic information. And this is where you're looking at normally naturally occurring mutants and trying to link that to a phenotype, or as time went on during the 60s, 70s, and even into the 80s, where you had mutagenesis studies where using chemical, radiation, or viral mutagens and looking at the mutations that are generated from that and the phenotypic effects of that. And that's, of course, is how actually the functions of many genes were discovered over the last few decades. But what we've been able to see really over the last 10, 15 years is move within a wider range of organisms the ability to do reverse genetics. And that's where you can use prior genetic information to generate reagents that then use to induce a phenotype. And these are important differences when you're thinking about it, because actually comparing and contrasting these results is still very valuable for many in looking at gene function. So I just wanted to clarify where we're looking at the application of these. For the purposes of this presentation, I'm going to exclusively focus on looking at transformed cancer cell lines and discussing it in that context. But I obviously want to make sure everyone's very aware of that now. A lot of these methodologies, particularly in some model organisms, have been extended to the whole organism, whether it particularly early forward genetic approaches in bacteria and in yeast. And then more increasingly, we've seen now we can do these analyses on whole organisms that are much larger, more complex, from C. elegans all the way through to mouse. But today I'm going to just discuss transformed cancer cell lines. But most of the methods I'm going to talk about are increasingly being applied in other types of model systems. And as you're thinking about how to apply using functional genomics tools, defining exactly which model you're going to use for applying these tools is very, very important. So I said before that loss of function comes in sort of two flavors. You have this knockdown, which is a consequence of a silencing or an interference type effect, or you have a knockout. So the classic form of gene silencing, this knockdown, and what I've been working on really now for almost 15 years, is RNA interference. Okay, where you're seeing more recently is where you have now gene editing tools, where you can, depending on how they're applied, you can have either a knockdown or you can have a complete knockout. For today's presentation, I'm going to focus on a discussion of RNA interference and then just actually these two forms of gene editing technology, predominantly tails or talons, and then more recently the CRISPR-Cas9 system. The application work I'm going to talk about, though, will only be in the context of RNA interference for today. Okay, so moving on to the technologies that we're using. This is just a sort of a historical context for RNA interference. So RNA interference, what we now know as RNA interference, was actually first described back in the early 1990s. And it was observations that were made actually, first of all, in these plants, which are actually petunias, and in the fungi Neurospora, that when you introduced plasmids that actually were overexpressing a cDNA, and where these had gone in as head to tail repeats, what you observed was actually a loss of the expression of the gene that was trying to be overexpressed. It was unclear what was causing this until the work of Andy Fire and Craig Mello in the late 1990s, where when they described in C. elegans that double-stranded RNA were able to induce this effect of gene silencing. I came into the picture in early 2000 with work establishing that if you now took small versions of these of double-stranded RNAs and introduced those into mammalian cells, you could induce the same effect through what we now know is and I'll mention very briefly in a minute, a naturally occurring gene silencing mechanism. What's been really exciting is that over the last few years, we've seen this move very rapidly into the clinic. I'm not going to talk about this today, but we have seen even clinical trials based on exploitation of this technology. And then I will talk a little bit at the very end about genome-wide screening using this technology. So the RNA interference pathway is a naturally occurring pathway. It's a very, very important one for the regulation of gene expression. And so in its naturally occurring form that every cell requires, this mechanism is processing a series of non-coding RNAs known as microRNAs from pol to express primary transcripts. These go through a series of different nuclease steps and are exported initially as a hairpin structure and are loaded into a Argonaute, a RNA-induced silencing complex that, if it's regulating gene expression, will normally interact with sequences predominantly within the 3' UTR of a target transcript through a binding at the one end of the molecule. I'm going to come into that in a little detail in a few minutes. This leads to mRNA degradation or translational repression. There's still, even after nearly 15 years of work, still some discussion as to which comes first. Certainly, both are probably playing a role. And this is a very precise and very heavily regulated and very essential process in regulating the expression of a very, between 30 and 50% of protein encoding genes, if not probably almost all protein encoding genes at some point. Now, interestingly, this had been observed for many years as present in C. elegans, but the pathway had not been really worked out, and it certainly had not been perceived that this was a more widely used pathway by cells. Until the observations that, in fact, we saw that in mammalian cells, you could actually exploit exactly the same pathway to induce gene-specific silencing of genes. And in this case, this is by using artificial mediators of RNAi that either mimic very closely what you see for this structure of a precursor microRNA, that you use a short hairpin structure, or you use an siRNA, which is a synthetic version of these. These will be introduced into the cells. They will access the pathway at this point. But what's different here is they will interact with the target transcript with 100% alignment of the sequences. And that will then induce a cleavage of the transcript and therefore loss of that transcript and therefore the loss of that protein. And that's sort of illustrated here. I don't know, it's not showing the cell on here. There's actually supposed to be a cell here, but it doesn't matter in this context. So what you're seeing here is you can use either siRNAs or siRNAs, and then after a period of time, having gone through this process, you will see a reduction in the transcript, reduction in protein synthesis, and therefore a loss of function. Now, my lab has predominantly, over the last few years, used siRNAs, and a lot of other labs have used shRNAs. But broadly, the advantages and disadvantages of these are really based on what it is that you want to actually experiment, how long your experiments need to last, and what cell type you're using it in. So siRNAs are a transient induction of silencing. This will only go from about 48 through to 144 days. It's a relatively short period of time. Whereas shRNAs, you can actually make stable cell lines. You can induce this, express these from plasmids, and make that last for very, very many passages, going for weeks if necessary. You can do genetic screening with shRNAs by doing shRNAs in pools, or you can do arrayed screens with sRNAs. The sRNAs, you have a lot more control over the system than you do with shRNAs, and so that's what we favored. But as I said, they are different people use them in different contexts. I think along the issues being is that in the context of hemopoietic cells, so T cells, B cells, you really have to use an shRNA because there's not an easy methodology for introducing siRNAs. So what is a typical RNAi experiment, or for that matter, really any typical loss of function experiment going to look like? Where you're inducing a knockdown. So if you're inducing a knockdown, what you're going to see, you hope, is a reduction in the levels of mRNA. In this case, we're measuring the Wnt signaling protein, beta-catenin. You're going to see a reduction, you hope, in the levels of the RNA with the introduction of the siRNA, usually after about 48 hours. Typically, that's what we would look at. If this is a knockout, then you would want to go down to effectively 0. You should not be having any production of transcript at all. But this is what we're looking for usually in this context of interference type experimentation. This should then be mimicked at a protein level. And here we're seeing again decreases in protein using the same beta-catenin siRNAs. And we're now looking over time. And we can actually quantify this if you have standard curves and you're doing this really carefully. We can quantify over time the amount of reduction that you see at a protein level. Now, the amount of protein decrease you will see will vary dependent on the half-life of that protein. These are not going to be linear responses. You may get transcript degradation very quickly, but if your protein has a very short turnover, you're going to see effects very quickly. If your protein has a very long half-life, then you may not see any effects at all. It may be very, very stable. And this can be, something you really have to think about when you're timing your experiments and when you're looking at effects. And I'll talk about that a little later in the context of one experiment we were involved in. So all the functional genomic technologies I'm going to mention this afternoon are exploiting the interaction of a sequence hybridization in some form, whether it's an RNA to RNA or whether it's RNA to DNA. You're using some forms of alignment of sequence. And in all of these approaches that are currently being used, there can be a varying degrees of tolerance of mismatches within that. And that will mean and can lead to what are thought of as off-target effects. So in the context of siRNAs, what you have here is that in most cases, your siRNA, this is going to have an exact match to the sequence that you're trying to target. But if you haven't designed your siRNA or your siRNA hasn't been designed very, very carefully, you can end up with key features of the siRNA sequence acting in a microRNA-like way because of the seed sequence at one end of the molecule. And that means then you can end up with a series of off-target effects where your siRNA is actually having an interaction with another transcript and may be either causing its degradation or is at least inhibiting protein translation from that target. Now, over the years, we've come up with lots of different ways to try and address this. Most of these are really quite basic. A lot of it can now increasingly be dealt with at the design end by just making sure that the seed sequences are as well defined as possible. Using a lowest concentration of the siRNA or any RNAi effector, or in fact, any of the reagents that I'm going to be talking about today, using the lowest concentrations of any of these is always a plus, and you really should aim for doing that. And you always need to know that you've used all the negative controls that you're going to require to be able to be sure that you've got an effect. In some cases, you do see in some cells an immune response. Again, this is rare, usually within the context of a standard lab experiment when you're into it. But there are some cell types, for example, some that do seem to be more sensitive to just the introduction of nucleic acids than others. They will trigger toll-like receptor signaling based on the fact that they've just seen nucleic acid. And then, as I said, we want to be able to be sure that we're using the lowest concentration to offset any unintended targets through partial complementarity here, negative controls. And then the other thing that we often do is we will use multiple different sRNAs that target different part of the gene that we're interested in. So you're using different sequences and comparing those phenotypic effects to give us more certainty, increase our certainty that what the effect that we have is real. So one of the groups that have evolved from my group, and I'll be talking about at the very end, is the Trans-NIH RNAi screening facility. They do functional genomic scale, genome-wide scale screening. And one of the ways that they've developed to try and counteract this issue of off-target effects is actually to make siRNAs where you actually mutate the middle part of the siRNA, which is absolutely essential for locating the cleavage event. And then you can compare the effects of your siRNA that's actually targeting your mRNA of interest versus the effect of when you have an siRNA that is mutated at that site. If you have an off-target effect and you see an effect that you on this mRNA that's not your target, in the context of these what we call C911 and C911 controls, then this is an off-target effect. But if this siRNA no longer works, then this is your on-target effect. And this can be very useful as an additional way of supporting evidence for the effect of your siRNA. The other method that I haven't written up here that people do use, though it's becoming less of the case you see in the literature, which is to do formal rescue experiments. Those are difficult to do, but are the definitive way of being able to establish that you have induced a loss of function and then you put back in the cDNA and can rescue that phenotype. So I'm now going to move on to a couple of other approaches that probably in the last 3 and certainly in the last 2 years have really come to prominence as being alternatives or complementary approaches for trying to induce a loss of function phenotype. So the first is to use tails, which are transcription activator-like effectors. So these are programmable DNA binding proteins that were first identified from the Xanthomannus gram-negative bacteria. These can infect a very wide range of plant species. And in fact, these DNA binding proteins appear to be part of their adaptive immune response. So the DNA binding domains that have been developed for loss of function contain 7 to 34 homologous direct repeats of a series of amino acids, usually around 33 to 35 amino acids. And these are designed to look at a specific DNA motif. Now, a lot of work has gone in to find out how these work, and it's now known that the key part of the specificity is within the 2 amino acids at this 12 to 13 within each repeat. And this is known as the repeat variable diresidue. So designing these is actually really quite difficult, and they tend to have to be very long. So if you're going to, each site in the genome needs between 500 and 7,000 amino acids. So these have not exactly taken off as an approach, but they are very effective if you can define one that works. Now, most you can put different domains on these to induce different effects, but most induce a loss of function effect when it's using an effector domain such as the coupled associated box or the CRAB domain. And when this is fused to the TALEN and you are looking at a site within the transcriptional or close to the transcriptional start site, this will now block transcription. So this is why it's described still as a repression. It's still a gene silencing. It's not absolute. You're going to get probably similar to what you see with gene silencing. And this is where it's still going to have some transcription. Now, again, this is where you have to design these very carefully, and you're going to need to test different sites within the transcriptional site to get the maximum effect of suppressing the expression from this particular transcriptional start site. There are some rules that say you want to avoid the occupied core promoter regions, or regions where they predict that there's going to be very high nucleosome occupancy, but that's really quite hard to design into the algorithm. So this has been quite difficult to do. The other feature is that in this case, these monomers, however long the sequences are, they still tend to mediate off-target effects. And so these are definitely, there's some great papers using these, but they've not become generally applicable. Now, what has been used perhaps more extensively is a version of these that actually now uses a dimer of these. So this is known as TALENs, because now what you do is you're adding to the tails a nuclease effector domains from a restriction enzyme, FOC1, and this is fused to 2 different tails. And by doing this, you're actually now targeting 2 different sequences. This means you've actually got increasing your specificity, because you've now got to have this molecule will only work if you end up with a dimer here, and they have to find 2 different places very close to each other. So this will then introduce DNA double-strand breaks, and then you've got 2 approaches that can make use of that event. You can induce homologous recombination repair, which if you add a DNA donor, you can introduce a specific point mutation, you can put in targeted deletions, or you can put in an insertion with a specific DNA fragment. Alternatively, you can use error-prone, the non-homologous end joining. This will allow for error-prone repair, which can give deletions, insertions, substitutes, lots of different frame shift mutations. Anything will, if you do it earlier enough in the gene structure, if you're doing this an early exon, will now result in a loss of function phenotype, because you can now no longer make the transcript. So this is a lot more powerful, because you having 2 more events and you have a lot more options as to how you're going to induce your loss of function. So TALENs have become quite more commonly used for this attempt, but I think we're going to see that this is sort of going to get superseded quite quickly by the new kid on the block, so to speak. So even my 13-year-old has heard of CRISPR, not from me. He's heard it because he's seen it on the CNN Kid News, because everybody's talking about CRISPR, and it will probably get the Nobel Prize next year. So I'm only going to give you the very edited highlights from this, because to be honest, I can't keep up with this literature. So if you're going to be interested, I'm just going to give you some highlights. Go read about this, because there's a new way of using this every single day at the moment. And so I just want to make sure everyone here who hasn't, if you've been hidden under a rock and haven't heard about this, then I just need to make sure in the process of this presentation you've all heard the basics. But please just go out from that. I've given within this a few citations of them, some of the more recent reviews that are really good, that just as a starting point for you. So like both RNAi and TALENs, you're using resources that have come from studying naturally occurring mechanisms. In this case, you're using it from a programmable DNA nuclease that's been isolated from bacteria. It's thought to be part of it's known to be part of their adaptive immune system. And the key protein is this Cas9, CRISPR-associated protein 9. So in most of the functional genomics world that's been adapting this technology, you're using it from Streptococcus pyogenes. And though I think you're going to see, well, I know you are seeing lots of different versions of this enzyme being generated that will have pros and cons and different aspects to it, until we settle on something that's probably going to be the most widely used. So Cas9 has innate nuclease activity. Okay. Now the targeting that uses this enzyme is achieved using a chimeric single guide RNA. It's known as an sgRNA, and this is usually thought of as a fusion of 2 parts: the CRISPR RNA and the transactivating or cRNA. Okay, so the constant region of the sgRNA that interacts with the Cas9, and there are defined sequences that are available, and those are really pretty well being sold now as a de facto sequence. You can the variable region is what is going to target the genome. That's the bit you have control over. That's the part that you're going to be using to edit your particular sequence of interest. And the genome targets usually is 17 to 20 nucleotides. So again, still very small real estate as a consequence. Now, the major specificity determined for Cas9 is actually a sequence that's adjacent to your target sequence. And it has to be immediately at the 3' end of the target system. And this is known as the protospace adjacent motif, or the PAM. And it's just for this species really just seen as 3 nucleotides, N, G, G. There are a few papers out there, and they're starting to see where, this is a moment, trying to find what's going to be the perfect PAM sequence. Where do you want to locate this? And eventually, a lot of those rules will be worked out. There's actually an incredibly cool paper that's in, I think it was in Science just this week, where you can actually they're doing live imaging of the Cas9 protein and looking at where it's binding and how it actually interacts with different sequences. All of this will be information that will be trying to make these tools better and better. Okay. So as with the TALEN, if you use a single Cas9 molecule at a very early exon, you're going to end up with, you can induce a random knockout as a consequence of a double-strand break and error-prone non-homologous end joining. But you can also get this in lots of other flavors. And I'm going to go through a few pictures here that will try to illustrate this. So this is showing the sort of typical model of what the Cas9 protein, where you have it. This is the PAM sequence on the DNA. And here you have the target of specific guide sequence that's going to match your target of interest. And here you have the standard, the consensus of sequence that interacts with Cas9. So this will induce a double-strand break, and then you have either non-homologous end joining, where you can get a small insertion or a small deletion, and if you choose it at a point earlier enough in your gene, you're now going to inhibit activity. But in the same way as I described for the TALENs a few moments ago, you can also get homologous recombination if you introduce a donor DNA that will allow you to get either insert in or a sequence or make other rearrangements, as I'll describe in a minute, because some of these are getting now quite sophisticated. So what I've just described now has been predominantly discussing really what is a knockout. So here are the different approaches where you get an insertion or a deletion. You can get an insertion or replacement, or a large deletion or a rearrangement using non-homologous end joining or homologous repair. But you can also use these in the same ways you do with TALENs and put these in combination with some sort of activating domain if you use a mutant version of Cas9 that is now catalytically dead. And you can actually now target the transcriptional start site, just as I said for TALENs, to get gene activation, or as I'll go into a few more details in a minute, to get transcriptional interference, or CRISPR-I as it's now called, which is sort of akin to RNAi, but using CRISPR. Probably one of the most important applications actually for Cas9 is actually the introduce and tag, how you can tag this with actually other proteins for imaging. And I'm not going to talk about it in detail, but that's a really useful way. And it's going to be both using it for imaging of the processes involved in for transcription, but also generation of cell lines is an important application for these technologies. So just for completeness, because we've been talking about RNAi and also TALENs, I wanted to just do one more slide on what's CRISPR-I. Now, this is only, I should stress, that there's a lot of reagents out of there. There's a lot of buzz about this. It's still going to be a bit of a work in progress to make this really effective and be able to be used quickly in the lab, I think. So this makes use of an enzymatically inactive version of Cas9 known as DCAS9. There's lots of these versions of this available. And in the case of when doing CRISPR-I, it's then fused to the repressor domain, usually of the crab bee, in the same way as it's used for talons. This can induce heterodomain mediated gene silencing if you're targeting the transcriptional start site. Now, you have to usually target a site that's about 500 base pairs downstream of the transcriptional start site, so you're avoiding where all the other proteins are so that it can access it. And one of the key problems with this, and it applies really to talons as well, is it assumes that every transcript from a particular gene is being expressed from the same transcriptional start site. And that's not always the case. And it may limit your ability to target lots of different variants of a particular gene. So that's just something that has to be borne in mind. It also may not work in regions that are with closed chromatin. So I think there's a lot of there's clearly a lot of interest in the community about being able to use this. We're certainly interested, but it's not quite a prime time for everybody to use at this point. But certainly, if you're interested, I think go ahead and give it a try. I mentioned with the other with RNAi and with talons that you do have risks of off-target effects. In the case of CRISPR, sorry about that. We know that it's the proximal 8 to 14 nucleotides that are immediately upstream from the PAM that are absolutely important for target site cleavage. And in fact, there's been thought that within that is a seed region in the same way as we think about it in the context of microRNAs. But it's clearly been seen that if you take dead Cas9, it will bind in lots and lots and lots of places. So it can cut in a lot of places. And that if you are using very high levels, particularly of the sgRNA, this could give you off-target effects. This is going to get started to work out over the next 6 months to a year, but it is something that everyone has to be really careful of. Mutations, one of the approaches that people are taking is if you mutate one of the 2 Cas9 nuclease domains, you can then make it a nickase. And then you have you have to have 2 nickases that are targeting 2 different sites. And then that will reduce your off-target effect, because if you have to have 2 cuts to get your effect, that will reduce your effect. So that's one approach individuals are taking to that approach. So I appreciate now that you can't read this, but I really wanted to just put this into to have individuals actually just look at this this review. This really summarizes some of the ways in individuals are using CRISPR Cas9 systems in cancer biology. Some of these are going to be the same as I'm going to talk about subsequently in terms of the applications of the work we've done in the past using RNAi. So they're really, I'm going to be using those as illustrative of any of the types of work people are using for loss of function, with perhaps the big exception of this application down here, which is now where you can actually potentially use, well, it's been shown you can use CRISPR Cas9 systems if delivered to, for example, the lung. You can actually use them to actually induce a generation of a translocation. You can translocate, and now you can make models of fusion-based tumors de novo, which has not been feasible before. And I think this is going to be probably one of the most exciting applications of this technology in the context of trying to understand some of the basics of cancer biology, though clearly a lot of these other approaches will be important. Okay. So in another review that I'd like you to just bring to your attention, this is perhaps maybe the most important take-home slide of my entire presentation. So where really Mike McManus and his colleague went through and did a really great job of trying to analyze the advantages, disadvantages, and put align the different technologies that are now available to you for loss of function analysis. And so they've just sort of summarized this in terms of the mechanisms, what you're going to see as a result of this, how you're going to induce this, and what is the likely off-target space within this. And as I say, at this point, they all have their pros and cons. A lot of it's going to be what your lab is used to, how much investment you want to put into one particular technology versus another, and how much it might be that you want to just wait until some of the newer technologies have matured a little more, or you just want to get in there and give it a go. So this is a really good summary of that. And if you're thinking about a specific experiment that you want to do, this is a good place to start if you want to try and compare and contrast the different options that are out there. So what I'm now going to do is just spend the last part of the presentation just going through a few examples of what my lab has done over the last few years using RNAi-based analysis. But actually, all of these are types of experimentation that can be done that would be equally applicable whether you were doing RNAi or CRISPR or any other. And I'm just going to run through a few examples just to give you a flavor of what can be done. So one of these, first one is was a kinome, actually just a screen of the tyrosine kinome using siRNAs, so that we were using this in a triple negative breast cancer cell line, MDMB231. We were asking a very simple question. When we silence a gene, what happens to these cells in terms of their viability? Basically, what genes are essential? What do these cell lines need? So we are ranking everything here versus the siRNAs against the ribonucleotide reductase subunit RM2, which we know is absolutely essential for cell viability. And the gene that was at the top of our list on this one with MDMB231 was this protein gene WEE1. So when we extended that to other cell lines, we saw that these cell lines were also, these are other breast cancer cell lines, these were also susceptible to silencing of WEE1. But the cell line MCF10A, which is a non-tumorigenic mammary epithelial cell line, we saw some reduction in cell viability, but it was significantly higher in the other cell lines, even though we've got similar knockdown at protein level of the protein. What was important is we could phenocopy this with a WEE1 inhibitor, which was seen here, and we could extend this now to multiple different breast cancer cell lines representing multiple different genetic variants of breast cancer. And still we saw no effect on the growth of these MCF10A control cells. We were able to show that WEE1 inhibition is actually causing the induction of DNA damage, potentially in just the breast cancer cell lines, and then we saw no induction in these MCF10As, giving us the basis for why we were seeing sensitivity in these cell lines, but not in these cell lines. And this was actually something that was confirmed in another study that came out almost at the same time as our study. Now, that all sounds great, and is something that has actually been presented repeatedly in terms of the cancer field over the last few years using RNAi to find new molecular targets for cancer. And it's something that's actually always concerned me because we actually know that WEE1 is required for the entry into mitosis through its interaction with the CDC2 serine threonine protein kinase. So this is is held in check by WEE1, and entry into mitosis is dependent on a protein phosphatase that removes this inhibitory phosphorylation from event. So WEE1 is actually required for mitosis in every single cell. So our first assessment, this is not unique to cancer cell lines. What we're seeing is that you have a window, a potential therapeutic window. But that is something that really has to be borne in mind as you look at a lot of cell viability-based screens that are done, whether it's done with RNAi or CRISPR or any technology. And that's really been borne out by two studies that came out in Science only in the last few weeks. In fact, I think they're only still on Science Express, where this was a CRISPR screen done by Eric Lander and David Sabatini. This was a mixture of different types of approaches. They used different approaches to induce, and where they were looking at what is really, truly essential genes. And if you actually go into this data set, WEE1 is essential, okay? So this has to be particularly borne in mind by the cancer community. So there is a huge literature out there that now says that a lot of proteins that we wanted to think of as having some selectivity, and there may be some selectivity, are not as selective as we thought or we wanted them to be. And so we have to bear that in mind. And this, I think, is going to be a really good data set for people to go back to and say, just remember that if you're pulling something you think is a hit, when you've used a very broad phenotype, such as cell viability as your endpoint, you may run into problems. So there are other approaches, though, for using loss of function analysis to understand the function of a particular protein in the context of cancer. And this is in the case where on colorectal cancer, a part of chromosome 13, 13q12, is consistently amplified in really the vast majority of colon primary tumors and in rectal tumors as well. But it was unknown what, if any, proteins are expressed from that would being deregulated as a consequence of that amplification. So I was involved in a study that focused on a number of the different genes that are present that map to this region. For now, I'm just going to focus on one of them, this gene here, LNX2. Okay, so one of the approaches we took to try and understand what is the consequences of LNX2 on tumorigenesis was to take a colorectal cancer cell line and then take 2 different siRNAs, silence LNX2 in these cell lines, and then look to the transcriptome of these cells. And we were able to then find the genes that consistently change as a consequence of the deregulation of this gene. This was to give us some sense of the functional space in which this particular protein is acting, but in as unbiased a way as we possibly could. So we got from this a couple of different observations. So it's been known in the literature that LNX2 binds to NUM and is part of Notch signaling. So we were very gratified that we found within that transcriptome that we indeed did see changes in Notch1, and we saw changes in Notch-targeted genes too, as well, that we were able to confirm this. But we were also intrigued that there was another transcription factor, TCF7L2, that was also deep down regulated in the context of LNX2 silencing. And in fact, when we looked at the enrichment of transcription factor signatures within the RNAi signature, we found that there was a huge enrichment for the downregulation of known targets of TCF7L2. And this is important because this is directly related to beta-catenin signaling, which is also a very well-established pathway that's disrupted in the context of colorectal cancer. And what we're able to show from this is actually that LNX2 does decrease the expression of TCF7L2 at an RNA and a protein level, and actually does affect its activity in a assay of Wnt signaling. So in this case, we were able to take what was our observations in tumors and now take them all the way through to a mechanistic application that links this one protein to actually 2 pathways that are known to be disrupted in colorectal cancer. We've done a lot of work, as have a lot of others, using different functional genomic technologies to look at how we can use those to make the existing drug, repurpose existing chemotherapeutic agents, and also find different ways of looking at chemotherapeutic agents. This is not a chemotherapeutic agent. This is actually a very unusual drug. It's a enzyme drug. It's asparagine synthetase. And I came into this project in collaboration with John Weinstein, who was then at CCR, is now at MD Anderson, based on a series of observations that they'd made using what now are considered very old-fashioned technologies, using early microarrays, that there was a correlation between the expression of this gene, ASNS, and the sensitivity of cell lines, particularly this part of the NCI60 panel of ovarian cancer cell lines, and that the more ASNS, the more resistant a cell line was to treatment with this particular enzyme drug, L-asparaginase. And if you had low levels, these cells were very sensitive to it. So what is L-asparaginase? This is an FDA-approved drug that's been used for more than 30 years for the treatment of acute lymphoblastic leukemia. It depletes the blood levels of the non-essential amino acid asparaginase by hydrolyzing the serum asparagine to ammonia, and that basically starves the cancer cell. So asparagine it's a non-essential amino acid, and so what we hypothesized was was that maybe the endogenous levels of asparagine synthetase, which is required for the synthesis of asparagine, was contributing to the sensitivity of these cells. And whether we could use this as either as a biomarker or some effect to understand the biology, because there's a huge variability in the sensitivity of patients to the treatment with this enzyme drug. So what you're seeing now is the 3 cell lines that we studied, who have varying levels of asparagine synthetase and have varying sensitivities to L-asparaginase. And what we're able to do is get very good knockdown at an RNA level. We're able to find a time where we got maximal effects on protein, and then we could do the pharmacology. And what we could see from here was that we were able to get a potentiation of the signal in all, or sensitivity of these cells in all cases when we silenced asparagine synthetase. So now we had a functional relationship between the levels of asparagine synthetase and sensitivity of this drug. We've now been able to show that actually what the RNAi is doing is not just suppressing the expression of the endogenous levels, it's also blocking the upregulation of asparagine synthetase that you see when L-asparaginase is actually added to. So you actually end up with an increase, the L-asparagine induction of ASA is suppressed, and you get a really much further shifting of the dose response, and therefore the sensitivity of these cells. So actually these findings have actually been issued as a patent. They've been licensed to a company that's actually developing them as part of a biomarker for the application of recombinant L-asparaginase for treatment in patients, because you really now know you only want to give this to a subset of patients that are expressing relatively low levels of N-asparagine synthetase, or if you can block that upregulation when they're exposed to L-asparaginase. So RNAi, or any functional genomics approaches, can be used for the analysis of drug activity, because it can be used to identify synergistic molecular targets. That will allow you to use lower concentrations of a drug. You might be able to overcome drug resistance. And it can potentially, as we've seen in the case of the asparaginase, you might be able to broaden the clinical applications to other cancer types. Okay? So I'm just going to do, in the last couple of minutes, just go through one final story and then lead into just some conclusions. And this final story is really where we were actually looking at that first point, was could we find, again, biomarkers or synergistic combinations to work with a different drug approach, which was the use of the TRAIL peptide. TRAIL is the tumor necrosis factor-related apoptosis-inducing ligand. I'm just going to refer to it from TRAIL from now on. This will trigger the apoptotic intrinsic pathway, and it's been really touted as a potential treatment in a number of different cancers, but its translation to the clinic has been difficult. And so this was a work done in collaboration with Stan Lipowitz in the women's malignancy branch now, where we designed a whole series of screens with different endpoints going along the apoptotic extrinsic pathway to understand if we could find different proteins that were required and would act synergistically with TRAIL. So this was a set of screens that were done at this stage, and the kinome and phosphatome plus additional genes, done in the absence of presence of TRAIL, and was again done in the triple-negative breast cancer cell line MDA and B231. And we decided to do the entire pathway going through caspase 3-7, caspase 8, and cell viability to give us a real sense of along this pathway. We were able to identify all the controls that were required for it, and I'm just going to focus here on the caspase 3-7 activation. We were able to find a gene that, when silenced, now completely blocks this activation, which is, of course, caspase 8. And then we could find it if we silence a known inhibitor of the extrinsic pathway, we could actually now increase caspase 3-7 activation. And all of these everything correlated. And that's actually shown here, that we could get a really nice correlation in the presence of TRAIL between these different screens, or good enough for this particular context. In particular, we saw really great, as we would expect, correlation between the caspase 3-7 and cell viability assays in the screen. So from this, we found 83 kinases and 4 phosphatases and 63 genes that were putative regulators of TRAIL using the caspase 3-7, which was an awful lot more genes than we were anticipating. But what was important was we found really series of genes that we would expect to be there. BCL-XL, BCL-W, and known inhibitors of apoptosis. So that was good. So we were getting that part of it right, but trying to move forward was a little difficult. That's just the confirmation there. So we ended up focusing on a few of the of an inner core, if you like, of proteins that all seem to link together through other proteins that have been defined as required for trail. And what we were able to show, perhaps most importantly, was when we were silencing a lot of these. Now, we were able to recapitulate the effects on MDMB231 cells, but we could now actually take trail-resistant cell lines and now try and make these sensitive. And in particular, using this, silencing this particular protein, ACTN4, we can now turn what is actually a very resistant cell line into a trail-sensitive line. And we can actually now mimic some of these effects with drug combinations as well. So this was a very successful screen, and we were able to get some real hits out of it. But actually, we found that trying to make sense of a lot of the hits was actually very, very difficult, and it made prioritization difficult. And so actually what we ended up doing was actually deciding we needed more data. And this was actually one of the, what was it now, 37 projects that have now been, have either been accepted to or completed the pipeline for the trans-NIH RNAi screening facility, which span out from my lab a few years ago. This facility is a core facility that makes use of arrayed siRNA screening. They're also going to be moving into other functional genomics tools over the next year or so. And this allows you to go from assay development all the way up to a full screening campaign where you are doing the entire genome. To do this, you need some really fancy equipment. This is it's a full robotic system. You have lots of different tools that are required for this because everything now has to be very disciplined and very, very well controlled. We're very fortunate that we can do now actually very complex phenotypes. A lot of high content imaging is now used, and a lot of people are doing very sophisticated assays in this system, much more sophisticated than I've just described. And we've done a range of project areas now. The highlights that have been published, for example, was one in, where is it, in Parkinson's disease, which came out in Nature about a year ago. We're about to publish one in Ewing sarcoma, which I'm going to talk about in the last two slides. But we've got them in everything from Ebola virus to NF-kappa B signaling. So what does a whole genome screen look like? I'm just going to show you one piece of data from a screen that I've been involved in, which was looking at the Ewing sarcoma, the protein responsible for Ewing sarcoma, a bone tumor. This is a fusion transcription factor, EWS-FLI1, using an assay that was developed by a then clinical fellow in the pediatric oncology branch. What we've been able to do is find proteins that are absolutely required for the activity of the EWS-FLI1 transcription factor. And what actually comes out of these types of data, and this is always the depressing thing when you're trying to sell this, and we just had this paper accepted. So this represents about 18 months' work. To get to this graph represents about 18 months' work. And what you're seeing here is a representation of the whole genome data. So you're looking at 90,000 plus data points within this. And what we're looking for here is that we know that if something is affecting the transcription generally, it goes across this axis, but we can find the proteins down here that are like mimic the siRNAs that target EWS-FLI1 itself are down here that are just required for this transcription factor. And as I say, we're very pleased that the paper was accepted actually on Saturday, so it's very good. So I think I've left myself a couple of minutes for questions, but you're also welcome to email me too. Thank you. Yeah. I'm sorry? So, okay, so you can take out the wild type protein, or you can, if you're really lucky, you can design effectors that would just take out the mutant. Now, usually in most of these studies, people are taking out the wild type protein to find out what it is that the wild type protein is doing. So usually to study the mutant protein, you'll do overexpression studies of the mutant protein in another system. Okay, so sRNAs in the clinic have been, the most successful to date has been in the eye, because that's an immune privileged site. It's been used to target VEGF and and epithelial growth within the eye, and that has started to go somewhere. You're seeing the development of sRNAs for respiratory syncytial virus, for RSV virus. So Alnylam is leading that effort, and that's going somewhere too. There's also a lot of excitement about using effectively what is the same thing, but using them for microRNAs and targeting microRNAs. So there's, a long time ago, I started my career as a postdoc in the gene therapy field. So I still tend to be a little wary, but it's exciting. I think it was very interesting. The Washington Post was back up last week talking about nucleic acid therapy. So it's coming back in. There's certainly been major investment in the last few years. We'll just have to see where it goes.
One last one.
Okay, so there really aren't very good reagents that will allow you to effectively transfect using lipid-based non-viral systems into lymphocytes. And using some places, some teams will tout electroporation type approaches, but those tend to end up with very, very high levels of cell death. So if you are investing a lot of work in handling lymphocytes, I strongly encourage and urge people to use viral-based technologies. They're much more effective. You're not going to be fighting the system there. We just haven't been able to crack being able to do easy transfection in suspension cells with lipid-based technologies. Okay.
Natasha, you really updated everything.
I did.
Thank you.
All right, let's switch this off for a second so that Kurt can come on. You hold it. All right. There you go. We got better.
Come on, Gert. Let him load you up here. So our next speaker is Kurt Harris. He's got his medical degree from the Kansas University School of Medicine. He then did clinical training in internal medicine at UCLA. And then he came to NCI. He's head of the molecular genetics and carcinogenesis section in the Laboratory of Human Carcinogenesis. He's received many awards, and I saw him speak once at the AACR meeting where he got the Princess Takamatsu Award. And he's also chief editor of the journal Carcinogenesis. So his title today, Cancer Epidemiology and Mechanistic Biomarkers of Cancer, Precision Medicine Strategy. Kurt.
Thanks, Terry. It's good to be back. So I'm going to discuss some of the principles of precision medicine, and let me make sure this goes. It's going to be largely in the context of lung cancer, but I'll say something about colon cancer along the way. The kind of medicine that I learned and many of my colleagues learned is traditional medicine, in which you take a history, in which you get information, the medical and family history and lifestyle, do physical examination, you order some tests, you come up with a differential diagnosis and treatment plan. So what's precision medicine? Well, precision medicine builds upon traditional medicine. It's multi-layered, it's individual-centric, and it's interconnected. So it uses an information commons and I'll go through this in brief but hopefully lucid detail, that uses various omics, and I'll describe a number of those. So the Information Commons, again, requires integration of all these omics along with clinical information, epidemiological information. This is big data, and requires biocomputing skills and a lot of effort. The result is the knowledge network, in which you have a molecular classification of individual patients, hopefully to improve their diagnosis and treatment and health outcome, clinical medicine, but also to guide biomedical research. And this biomedical research feeds back into the Information Commons and also guide prevention research. So what's the exosome? It was coined by a fellow named Chris Wild, who's director of the International Agency for Research on Cancer. And you're all familiar with the environment and carcinogens that might be in the environment, tobacco smoke, infections, so on and so forth. But it also includes the internal environment that is directly influenced by the external environment, such as obesity and chronic inflammation, leading to cancer biomarkers of risk and prognosis. Genomics plays a role, both genomics and epigenomics, genetic susceptibility that might be inherited, driver genes that may be targets for cancer therapy, but this information leads to an understanding of carcinogenesis, how normal cells become cancer cells, and adds to this knowledge to develop cancer prevention and screening strategies. So one of the first examples of a connection between exposure to the environment, to carcinogens and molecular changes came from studies of going back many years ago, before we knew about molecular changes, of chimney sweeps having increased risk of scrotal cancer, studies that I've and other people have done on Aspergillus flava, increased liver cancer, and then cigarette smoke. Cigarette smoke contains about 65 different chemical carcinogens, and it's a witch's brew of carcinogens. As I mentioned, there's now, excuse me, now evidence that these carcinogens can in fact cause specific mutations in genes, including the P53 gene. So aflatoxin exposure, this was the first one that we examined, causes mutations in the P53 gene at codon 249. So there's a molecular link between the environment and, in this case, a tumor suppressor gene. Tobacco smoking, which contains many polycyclic aromatic hydrocarbons, primarily G to T transversions, and at multiple sites. Ultraviolet skin causes very unique CC to TT mutations, primarily at 2 sites. And this dietary contaminant has been recently shown to cause renal cancer, and it causes AT mutations with strand bias. So does this make any difference? Well, with P53 mutations, it does. So this is a study that we recently published with Snorri Thorgerson and Xin Wang, showing the prognostic values of P53 mutation sites in hepatocellular carcinoma. So if the P53 gene is wild type, there's good prognosis. These are called Kaplan-Meier plots. So this is 100% survival. This is 0% survival, and 60 months is 5 years. So mutations do make a difference, but mutations make a difference particularly if they're so-called hotspot mutations, gain of function mutations. And the 249 mutation is one of those, as well as the 157. And then if you compare wild type versus these non-hotspot mutations, there's not significance, but the hotspot mutations, there is, and that's because it loses suppressor activity and gains oncogenic activity. So I mentioned inflammation just a couple of minutes ago. Inflammation can be largely inherited, such as hemochromatosis, that's an iron overload condition, or increases risk of liver cancer, or inflammatory bowel disease, such as Crohn's and ulcerative colitis, and familiar pancreatitis. President Carter's family is a good example of familiar pancreatitis with a high risk of developing pancreatic cancer. In fact, most of his siblings and relatives have died of pancreatic cancer. He is one of the exceptions in the sense that he's in his 90s now and doesn't have pancreatic cancer. Unfortunately, he does have melanoma, which looks like it's being responsive to therapy. Most inflammation is due to acquired agents such as viruses, bacteria, parasitic agents that increase the liver cancer, gastric cancer, ovarian, and so on and so forth. And this is about 2 million human cancers per year are related to infection. And this comes from the World Cancer Report that I and many other people contributed to, that was published about a year ago. What isn't in this report are chemical, physical, and metabolic conditions that also increase inflammation. So acid reflux, esophagus, Barrett's esophagus, an increased risk of esophageal cancer, obesity at multiple sites. Obesity is a chronic inflammatory disease that increases the risk of cardiovascular disease, neurological disease, and multiple types of cancer. And then smoking. Now, I just mentioned that cigarette smoke contains about 65 known chemical carcinogens, but it's also a very potent inflammatory agent. Now, I want to ask which of you actually have smoked or have taken a draw of a cigarette, but the very first one you take is very toxic and usually makes people cough. And if there wasn't nicotine in tobacco, no one would be smoking. I can guarantee you that. Unfortunately, that's not the case. So these other causes of inflammation are associated with many more cancers per year. And for example, smoking, about 6 million cancers per year in the world. It's the leading cause of cancer in the world by far. I mentioned obesity. Now, there have been two very important observations or periods of observations on obesity. One was about 2003, in which two of these papers described that the fact that there are macrophages that have invaded into the body fat and form positive feedback loops with pro-inflammatory cytokines. The second is a more recent couple of papers in which show that adipocytes, fat cells, can senesce. And why is that important? Well, senescence is important because senescent cells produce a lot of inflammatory cytokines, such as IL-8, IL-6, TNF-alpha, and beta, and NF-kappa B pathway inducers. So you have these pro-inflammatory loops that give rise to a chronic inflammatory state. There's also free radicals that are formed. So let's look at some data. These are some studies that we've done asking the question, can one measure in serum inflammatory cytokines, and are they predictive of risk, diagnosis, or prognosis of lung cancer in this case? Well, for increased risk, one has to do prospective studies. So this is a study that was started about 35 years ago, and samples were collected, including serum. And then over that 35 period, this was almost 100,000 people, there were individuals who developed different kinds of cancers. So then you can take that group of those individuals and controls and do a nested case control study and go back and look at the serum that was collected 20, 30, 40 years ago. And when we did that, we found that two cytokines, C-reactive protein and here we go, and IL-8 were increased prior to diagnosis, including up to 8 years prior to diagnosis. So how about at the time of diagnosis? Well, IL-6 joins IL-8 at this point, both in Afro-Americans and European Americans. We're quite interested in health disparity kinds of studies, but this initial prospective study had only about 5% Afro-Americans. In our later studies, we've been able to look at about equal numbers of European Americans and Afro-Americans. However, the Afro-Americans had two other cytokines that were increased, interleukin-1 beta, which is a pro-inflammatory cytokine, and IL-10, which is an anti-inflammatory cytokine. How about survival? Well, one can predict survival by measuring these various cytokines, and I'll show you that in a little more detail. So there's poor survival in lung cancer, including early stage lung cancer, if IL-6 and IL-8 are increased, and if there's an increase in mannose binding protein, which is in fact involved in innate immunity. And this is a genetically inherited condition. About a fourth of us sitting in this room have an increased amount of mannose binding lectin type 2 in our blood. And again, Afro-Americans have high levels of IL-10, but also high levels of IL-12. So I mentioned that even in early stage cancer, and this is stage 1A lung cancer, so these are tumors that are no bigger than this, and there's no evidence of metastases after surgery. But as you can see in this Kaplan-Meier plot again, 100% survival, 0% survival, and up to 15 years, that if both IL-6 and IL-8 are low, in fact, survival is quite good. It's about 80% over this period of time. If one out of the two is high, survival is about 50% over a 5 to 10 year period. But if both are high, that's the red line, then only about 70% of the individuals will survive. Excuse me, I put the other way. 30% of the individuals will survive. So this is associated with increasing risk of recurrence and death. Now, a number of years ago, in fact, a long time ago, I was at a meeting in Cape Sunion, Greece, very scenic spot, quite different than it is now, I might say. And at that point, the late Hirayama, Professor Hirayama, presented his first evidence that passive smoking or environmental tobacco smoke increases the risk of lung cancer. Study done in Japan. And I must say, there were very few people who actually believed him. But he was absolutely right. There are 50, 60, 70 papers that have confirmed that. So it's quite well established now that environmental tobacco smoke, the smoke that you might inhale from someone else's cigarette, increases your risk for cancer. And of course, Hippocrates' disease comes from Greece, and this is one of his translations. And it goes, some men have constitutions that are like wooded mountains, running with springs, others like those with poor soil and little water, still others like land rich in pastures and marshes, and yet others like the bare, dry earth of the plain. So this is a long time ago, 25 centuries or so ago, and at that point, there was very little that one could do in diagnostic tests, except one, which I'll show you in a moment, but you focused on the individual. You focused on the patient. And good physicians still focus on the patient. They don't sit at their computer with the patient in the room for 8 out of the 10 minutes that the patient is in the room typing things. They actually talk to the patient. They put their hands on the patient. They give advice to the patient. A bit old-fashioned, but that's what I am. All right, so as I was sitting at this meeting, I was thinking, well, how could I actually investigate what Hirayama was reporting? And so I thought, well, maybe look at never smokers, who about 10% of the people coming in our clinic are never smokers now. That was one. Secondly, wait until there was enough time for lung cancer to occur in people whose mother and father smoked. And most of the cases, the mother smoked during pregnancy. This started in the Second World War here in the United States. So I had to wait until about 2007, 2008, and Susan joined the lab at that point. And the hypothesis was that childhood exposure to secondhand smoke and genetic alterations in innate immunity increased risk of lung cancer in never smoking adults. And this actually showed to be true. So parental secondhand smoke during childhood is associated with a dose-dependent increase in lung cancer risk among never smokers in 2 different cohorts, a Mayo Clinic cohort and a cohort here in Maryland. And this is especially true of those who have this genetic predisposition and a hyperactive innate immune system from mannose binding lectin-2. What was really surprising to us was that these lung cancers occurred primarily in people in their 50s. That's quite early. Most lung cancer occurs in the 60s and 70s, even in active smokers. So there were an increased risk. There's a genetic component to this predisposition, and secondly, their cancers occurred earlier. So let's go to genome and lung cancer. So these are mutations primarily in genes within the genome. So lung cancer, traditional view was small cell squamous adenocarcinoma. In '87, KRAS mutations were first detected. In 2004, EGFR mutations were also detected, and that led to the first therapy directed toward EGFR kinase activity. And last year, the TCGA, that's the Cancer Atlas that you can look at, sponsored by the NCI, identified a number of additional sites of mutations, primarily in never smokers, not in non-smokers, I mean in current smokers. And there are a number of drugs now that are directed toward these various activated kinases and like ALK kinase. The 2015 version of this will be coming out soon. It includes a few more sites, and it includes for the first time gene amplification of a microRNA, one that we're studying and I'll be talking about in a moment, called miR-21. So now let's go to the transcriptome and the epigenome. And microRNAs. So microRNAs are small non-coding RNAs. They're about 24, 26 base pairs, so they're quite small. And they were discovered by this fellow right over here, Victor Ambrose. Now, Victor is a basic scientist who was studying C. elegans, and that's a yeast. And he made the discovery that there were these small RNAs, and they had function too. Well, this is an hooray for basic science, but this was a new discovery of something that was evolutionarily quite old. So plants have microRNAs. And they regulate gene expression by either binding to the message and inhibiting the translation of the message, or binding to the message and causing instability of the message. And they affect hundreds of genes. Each one affects more than 100 gene expressions. So you just heard about siRNAs and the fact that you really work hard when you're in the laboratory to not have any off-target effects. So you look at multiple siRNAs or shRNAs, and you find those that seem to be affecting the gene expression and that you're interested in. Well, that's great for the laboratory, but this evolved not in that way. It evolved in a way, the microRNAs evolved in a way that they have multiple targets. They affect them in multiple pathways. Some are in the same pathway, the targets are in the same pathway, and it's not on or off in terms of their effects. It's modulating the effects. Well, this became exciting to the field of medicine when this guy here, Carlo Croce, a collaborator of mine, made the discovery of that 2 microRNAs were deleted, miR-15 and 16, in chronic lymphocytic leukemia. This is 2002. This is a seminal discovery in medicine. Since that discovery, there are tens of thousands of papers that have studied microRNAs, not only in cancer, but cardiovascular disease, neurological disease, rheumatic disease, all kinds of different categories of disease. Well, that led to us and many other people to ask the question, are they differentially expressed in human cancers? And can they predict risk, diagnosis, prognosis, and therapeutic outcome? What's going on there? There we go. All right, so I mentioned that these small microRNAs bind to message and can inhibit translation, but they can also bind to message to cause cleavage, and there's usually a base pair or two that is different in these targets. But microRNAs can also bind to proteins, including RNA binding proteins, and affect their function. And lastly, they can act as ligands. So they can bind to toll receptors and activate IL-6, which you've already heard about, and TNF-alpha. So let's start looking at lung cancer. And Nozomi was the one in our group that initiated these studies. And the hypothesis was that microRNAs are associated with lung cancer diagnosis and prognosis. And he actually found that the profiles were different between primary lung cancers and corresponding non-cancerous tissue, matched pairs, and among different histological types. So you could determine that they were squamous cell or adeno or a small cell lung cancer. He also found that there were 3 microRNAs that were increased, miR-21, miR-155, and miR-106, and there was a decrease in LET7. And this is, again, in the very earliest stage of lung cancer, stage 1. That led us to do a collaboration with the Croce lab to ask the question, well, of the major types of human cancer, which of the microRNAs are mostly overexpressed? So we did a fairly large study of breast, colon, lung, pancreas, prostate, and stomach cancer, and analyzed at that point, there were only about 280 microRNAs known. There are about 2,000 known today. And we found that miR21 is overexpressed in all 6 of these major types of cancer. That was 2006. And since then, it's been shown by us and others that it's upregulated in 18 major cancer types and is a biomarker of poor survival in 14. So this is a study looking at 3 different cohorts of lung cancer cases, a Maryland cohort, a Norway cohort, and a Japanese cohort. Again, these are Kaplan-Meier plots, 100%, 0% survival. 5 years is out here. If high levels of miR21, that's the red line, poor survival in each of these cohorts. And that's a principle of doing these kinds of studies. You don't rely on one cohort. It's important to look at multiple cohorts and, if possible, looking at geographic as well as racial and ethnic differences. So I mentioned there are 14 studies that have been shown that miR21 is a prognostic indicator. This is just about 10 or so of those that have been published. And I will say that each one of these has been verified by multiple other studies. So that's another important principle in doing these kinds of studies, is to show that they are reproduced in other people's hands. And we also studied colon cancer, and 2 different cohorts again, a Maryland cohort and a cohort in Hong Kong. And miR21 was associated with poor prognosis in both of these. And at that time, there were 3 others, actually after that, there were 3 other studies that showed exactly the same thing. Now, colon cancer is treated still today by 5-FU. There are other drugs that are used, but 5-FU is also used. This is a fellow named Charlie Heidelberger, who 50-some years ago discovered 5-FU. He also was a cell biologist and someone interested in chemical carcinogenesis as well as a pharmacologist. So a couple of years ago, we did a study comparing the effect of miR21 on the therapeutic response and survival of 5-FU-based therapy in 2 different cohorts, again, a Japanese cohort and a German cohort. And in both cases, if miR21 was high, there was poor prognosis associated with 5-FU compared to 5-FU if the miR21 was low. This is below the median. And this is also true in the German cohort, shown here. So I could spend an hour talking about miR21, but this just summarizes the key points. I've already mentioned that it could be amplified. It's frequently amplified in human cancer. It's regulated by DNA methylation. We showed that EGFR or KRAS will lead to an increase, that David Baltimore and his colleagues showed that IL-6 or interferon acting through activation of STAT3, it's phosphorylated, acts as a transcription factor for miR21. And in collaboration with colleagues here, Jim Mitchell and Nicole Simon, we showed that ionizing radiation and free radicals, in this case hydroxyradical, that increase miR21. Now, if you remember, microRNAs lead to a decrease in the amount of protein by those 2 different mechanisms. So these are just some examples that have been verified in the laboratory of critical proteins involved in cancer that are decreased if there's high levels of miR21. And as you can see, this is just a partial list of those. And also that miR21, as I mentioned, is a ligand. It's involved in the NF-kappa B pathway and also in cachexia, and I'll tell you more about cachexia right now. So these are, this cartoon is of 2 different studies showing cancer cells producing exosomes. So these are very small pockets of membrane containing cargo, including microRNAs. And these are about 100 or so nanometers in size. They're very small, but they're secreted by, if you want to use that word, by cancer cells and many other cells too. And these pockets of exosomes can fuse then with macrophages and lead to, in this case, miR-21, activating the NF-kappa B pathway to produce IL-6 and TNF-alpha, and perhaps a growth, tumor growth, metastatic potential. But Croce has just recently shown that miR-21 also in these exosomes will also bind to muscle cells. So what's cachexia? Cachexia is a condition that's relatively rare in cancer patients, more common in pancreatic cancer patients and perhaps some others. And I've taken care of 3 cachexia patients when I was taking care of patients who had lung cancer. And they waste away right in front of you. They lose their body fat, they lose their muscles, and die. It's a pretty terrible way of dying. We have no effective therapy right now for treating cachexia. But there are companies that are interested in using anti-MIR therapy for treatment of cachexia. And hopefully within the next year to 2 years, there will be a therapeutic trial done primarily in Brazil. All right, now let's look at protein coding genes and their expression. And this goes back to colon cancer again. And the hypothesis was the inflammatory risk score in tumor and adjacent non-tumor tissue correlates with colon cancer-specific mortality independent of tumor stage. So if you look at the adjacent non-tumor colon, we found that PRG1 and interleukin-1, interleukin-10, and CD68 were associated with poor prognosis. And in fact, IL-10 was associated very well with nodal metastases. So again, IL-10 is an anti-inflammatory agent. The colon cancer itself had these 6 different proteins that were overexpressed, including IL-23 that stimulates Th17 cells to produce IL-17, which usually is associated with poor prognosis. Well, then we ask the question, can you combine biomarkers that are mechanistically and statistically independent of each other? What I just said is very important. Mechanistically independent and statistically independent of each other. So we've done a number of these kinds of studies. This is one of colon cancer. The inflammatory risk score that I just talked about, if it's a high inflammatory risk score, prognosis is poor. If miR-21 is high, prognosis is poor. If both are high, prognosis is very poor. And if both are low, prognosis is quite good. And if either one is high, it's intermediate. And statistical analysis has shown that, let's see if I can get it. There it is. Now that's not good. Sorry to waste your time doing this. All right, we're back on track. So this is doing multivariate analysis, which allows you to ask the question, are the covariants independent of each other? In each case, in this case, it is that inflammatory risk score is independent of miR-21. and they're both independent of each other and tumor stage. Is this true only in colon cancer? No, it's true in esophageal cancer, lung cancer, and breast adenocarcinoma. So this is sort of a general principle. I keep doing that. I keep doing that. See if I can see this back over here. Okay. Well, that's going backwards. No. So can one do these kinds of studies in lung cancer? And we'll find out if this continues to work or not. I'm sorry. Better if I look down here. Yeah. All right. So this is a lung cancer study. Yeah, thanks. You would think I never gave a lecture before, wouldn't you? Interesting. Okay, so the hypothesis is that combination of miR-21 and 4 coding gene, in this case XPO1, that's involved in transport of microRNAs into the cytoplasm, BRCA1. Say, well, BRCA1 happens to be a tumor suppressor gene. That's true. But if it's overexpressed, it will cause genomic instability. HIF1 alpha, well, that's hypoxia, isn't it? HIF1 alpha is the protein is made primarily at the time of hypoxia at that level, not an increase in expression of the message. And then DLCA1 is in fact a tumor suppressor gene. So the 4 gene signature, protein coding gene signature, poor prognosis here. miR-21, if that's high here, and this in the Japanese cohort, primarily stage 1A lung cancer, 3 centimeters and less. The two together, poor prognosis. Well, that's backwards. I'm doing it again. We'll just use this. All right, so in the second cohort, in which primarily stage 1B, that's 5 centimeters and less. So the prognosis is even worse. So you can see the high, poor prognosis, miR-21, poor prognosis. So if both the 4 gene signature and miR-21 are low, very good prognosis. If either one's high, poor prognosis. And if both are high, only about 20% of the people actually survive. So what's the principle here? The principle is that non-coding RNA and coding RNA each are mechanistically and statistically different from each other, and they give you a better indication of it, whether it's a cancer or not in prognosis, but they misclassify some cases, but they misclassify different people. So that's the principle. All right, now let's end up with DNA methylation. And this is Anna Robles, who asked the question whether or not you combine the protein coding genes, the message that produce message, with microRNAs with DNA methylation biomarkers. And a study in Spain had shown that there are 5 different DNA methylation markers that are associated with poor prognosis in lung cancer, stage 1 lung cancer. When we looked at this, we said, well, HOXA9 looks like it's better than these, but are they dependent or independent of each other? And all 5 are dependent on each other. So we selected only looking at HOXA9. And when we did that, and we combined them with the protein coding genes, the microRNA, and then HOXA9, we found that if all 3 of these were essentially negative, very good prognosis in stage 1 lung cancer. If 1 is positive, somewhat poor prognosis. If 2 is positive, more poorer yet prognosis. And if all 3 are positive, quite poor prognosis. So that in stage B cancer, this has gotten out of place. These people only, the median survival at 50% was at 27 months. So metabolome. So that's actually asks the question, can you measure tumor metabolites in body fluids? And that's metabolomics. And unbiased metabolomics, the hypothesis would discover biomarkers associated with risk, diagnosis, prognosis, therapeutic outcome in lung cancer. And these are the 2 people who did these studies. We focused primarily on urine. And in urine, one can measure about 1,500 metabolites. And these metabolites are 1,500 molecular weight and less. So this is another history lesson. So as I mentioned, Hippocrates had only 1 laboratory test. And that was, at that time, tasting urine. Now, I never had to taste urine as a clinical test. I must say I tasted urine once when one of our boy child was very small, in diapers. And I got, well, you can imagine what happened. Anyway, this is a picture, an old picture, but this is a physiologist who wrote in fairly flowery language like Hippocrates. From a liquid window through which physicians felt they could view the body's inner workings, urine led to the beginnings of laboratory medicine. So we published last year a study of metabolomics looking at urine, and in an unbiased way, but 4 of these metabolites were associated with poor prognosis, and all 4 of them were independent of each other. If you put them together, the prognosis is, after a 5-year period, only about 20% of these individuals were still alive. And to show you that these are tumor-based metabolites, creatine riboside is positively correlated in the cancer, so it's high in the cancer compared to the non-cancer. And there's a correlation, positive correlation between the amount in the tumor and the amount in the urine. So that's this line right here. So let's end up with focusing on really an important clinical problem, and that's lung cancer and lung cancer screening. So this is another one of these reports, Surgeon General report, again, that a number of us contributed to. And this was 50 years after the first Surgeon General report. And they asked the question, how many premature deaths have been associated with smoking over that 50-year period. The bottom line here is over 20 million people have died because of smoking. And not only for cancer, but also cardiovascular disease and other kinds of conditions. Lung cancer in the United States is going down in males, and it's leveled off in females. We'll see what happens over the next few years. But we've been very successful in decreasing smoking in the United States. Only about 18-20% of the people smoke today. We used to be like this in China, where I was about a week ago, less than a week ago. I'm still jet lagged, in which 35 to 60% of the people smoke. We used to look like this. This is how Asia looks today. And so you can imagine the increase in lung cancer and smoking-related diseases that are going to occur in these populations. Lung cancer is already the #1 cancer. It's more people die of lung cancer than the 3 other cancers after that, cancer deaths after that, worldwide. So this is a serious problem. So how about screening? Well, this is a landmark paper that was published in 2011 in the New England Journal of Medicine, and they compared X-ray, which we knew didn't work very well, with low-dose computer tomography, or helical tumor computer-based tomography. And they found that there was about a 20% reduction in mortality. So that was good. And when cancer was determined, it was these early stage cancers, the ones I've been talking about, stage 1A, 1B primarily. What isn't good is the false positive rate is 96%. So that's even higher than the false positive rate with mammography. Mammography, the false positive rate is about 90%. So 10 biopsies in mammography, only one's going to be cancer. Another way of looking at this is, so these are 1,000 seats in an auditorium. And this is the total number of lung cancers that are averted by spiral CT scans or diagnosed. And out of 1,000 people that were screened over a 5-year period, there were three people who actually were diagnosed with lung cancer. But look at all the people that had false positive rates. And this causes a lot of anxiety and lots of overdiagnosis. And in surgery today, or in the community, the medical community, this overdiagnosis leads to many more surgeries than actually are needed. In fact, only of those people who go to surgery, less than half actually have lung cancer. A benign lesion is removed. Well, low-dose CT is from 2011 is recommended as a screening. Medicare started paying for it in 2015, and Affordable Care, Obamacare, if you want, if you're a Republican. I'm not. Is associated with paying for lung cancer screening. And the criteria is changing, but these are the more or less the basic criteria. And there's a need to identify people who are outside of this criteria. So I mentioned about 10% of the people coming in the clinic are never smokers. they would never be screened. And also, there's about almost 9 million people that are eligible for screening now. We don't have the capacity and won't have the capacity to screen that many people for quite a number of years. So there's also a need to prioritize those who are at greatest risk for screening, something that would be economical to do it. And some of the tests that I've been talking about in biomarkers perhaps will help with that. So in summary, stage 1 lung cancer after curative surgery, about 25–30% of those people die. Can be either stage 1A or 1B. These are low-risk people, and there are also high-risk people, and people who have occult metastases. So the hope is, and our objective is, to use precision medicine to decrease false positive rate, reduce financial cost, improve patient care, and guide mechanistic studies. And one of the questions is, in these high-risk people who now don't get any kind of adjuvant therapy, will immunotherapy and or chemotherapy improve their risk of recurrence? Are micrometastases from these early stage cancers, are they more sensitive to chemotherapy and immunotherapy than when you already have a large metastatic tumor? So those are the interesting questions. These are the people in the group that have done these kinds of studies. We've collaborated with a lot of people. I barely got through that today, but thank you very much. Any questions? Yeah, in the back, all the way back first. Okay, say that again. Oh, companies that are doing biomarkers? Yeah. So we've been talking to two different companies that are interested in pursuing the intellectual property that the NIH has, and we'll see how that goes. The biomarker field is one in which requires what's called CLIA-based assays, very standardized assays. Most clinical labs most laboratories, probably the laboratories you work in, do not have standardized tests that are required for patient care. So reproducibility in many of these studies, biomarker studies, has not been very good. And that's why I emphasize looking at multiple cohorts and having the results verified by other groups. And we have CLIA-based assays in our group. And as we develop a new biomarker, we also develop a CLIA-based assay. So in the stimulus to increase this with the screening, low-dose CT, is really driving the interest of the diagnostic/pharmaceutical companies, because if you can make the diagnosis better of who's going to have poor risk, and that company is also doing therapy, then that's in the interest of that company overall. And the companies we're talking to are, in fact, those who want to see more stage 1 cancer patients put into clinical trials. Yeah, Steve? Yeah.
So microRNAs are obviously a biomarker for cancer progression and whatnot. Are they detectable in urine versus serum samples?
They're detectable in serum and plasma samples. We've done those kinds of studies, and other people have done those kinds of studies. Plasma is probably better than serum because serum, there are microRNAs in platelets, for example, and in white blood cells. So serum comes from coagulated blood, so all those microRNAs are spilled out into the serum and can swamp the signal from the cancer-derived microRNAs. Urine, that's been studied in bladder cancer, and so that makes sense that you can detect microRNAs in bladder cancer that are, so that's being actively investigated. Good question. Yes?
So you mentioned the high false positive rate.
Yeah.
Unnecessary surgery. I'm wondering what are the steps in between that happen before the surgery and after the scan and breast to prevent those surgeries?
Well, there's several things driving the surgery. One is uncertainty. So physicians obviously don't want to miss a cancer, but they would like to have a better indication whether or not it's cancer or not a cancer. And even a biopsy is not always diagnostic. And secondly, some of the small lesions are in the periphery of the lung, and through a bronchoscope, it's hard to actually reach them. So, but if there could be serum or urine tests that say this is a high-risk individual, you can't say with absolute certainty, then most physicians would be happy to have that information. And if it was at low risk, they would be happy to just continue screening every 6 months or so. There's another aspect of this. People make money by doing surgery. Did I say that? Did I say that, Terry?